So with that, I just want to welcome our hosts, or our presenters today, Dr. Evgeny Zdobnov and Dr. Rob Waterhouse from the University of Geneva Medical School and Swiss Institute of Bioinformatics. They'll be speaking today about OrthoDB, so I'll let them take it away. Thank you so much.
Thanks, Anna. And for those of you who are calling in and can't see the slides are already up on the webinar website, so you can download those and follow them that way. So many of you are probably already familiar with the concept behind OrthoDB and what we provide because of the many different arthropod genomics projects that we've been involved in. But today I just want to spend a little bit of time, first of all, on the background of what we mean by orthology, and then go into a few of the details behind how we actually go about delineating orthology, and then finish off with the main focus of the presentation on looking at some of the features that OrthoDB offers to large-scale users, but also just individuals browsing for their favorite gene. So in terms of what is orthology, we'll have a look at some of the underlying definitions. In terms of the methodology, we're just going to briefly skim over some of the main ideas to help you get to grips with what's going on behind the scenes. And then, as I said, we're going to go into a little bit of more detail of how you can use orthology resources in your own research. So to start off with, what is orthology? To do that, we really have to take a step back and look at the parent term of orthology, which is the definition of homology. So orthology is really a child concept of the much more broader term of homology. Homology simply designates the relationship of common descent between any entities without any further specifications of the particular evolutionary scenario. And here I refer you to this Annual Reviews in Genetics article by Kunin, where he goes into some detail about orthology, paralogy, and the implications for evolutionary genomics. So orthology, being a child term of homology, is a more specific definition. And in terms of genes, it means genes originating from a single ancestral gene in the last common ancestor of the compared species. And for those of you who've been paying attention, you'll see that this review article also refers to paralogs. So what are paralogs? Paralogs are also a child term of homology. Here, genes that originate by gene duplication. So homologs, orthologs, and paralogs. Homologs simply refer to the common ancestry of any genes, whereas paralogs refer to a gene duplication event, and orthologs refer to a speciation event. And paralogs and orthologs are child terms of homology itself. We'll start off with a very simple scenario with 6 species that we all know and recognize, with a single copy ortholog in each of these species. This means that the last common ancestor of all 6 species had 1 copy of this gene, and the only evolutionary events that have occurred since their last common ancestor are speciation events, resulting in a traceable evolutionary history of that single gene in the last common ancestor to a single copy in each of the 6 extant species. A really simple scenario, easy to understand. Of course, evolution isn't always so simple, and other things happen. One event is gene losses. So here in the duck, we've lost this gene. The evolutionary history of the whole set of genes remains the same, just with the loss in 1 species. Other events, of course, include gains. So gene duplication events here in the human. So we have 2 copies, and in human, genes 1 and 2 are referred to as paralogs. Still a relatively simple scenario to get your head around, the evolutionary history of this gene family. Then, of course, you can have older gene duplications in a common ancestor intermediate between the extant species and the last common ancestor of all species, resulting in paralogs in both the rat and the mouse genomes. So you can see how, with different evolutionary events, our starting point of a simple scenario that just included speciation events can rapidly get much more complicated. So looking at different events, including speciation, gene duplication, and gene loss, across a phylogeny, we can begin to understand what we mean by orthology. We started off by firstly saying that it's a more specific definition of the general term of homology, which is simply recognizing similarities as evidence of shared ancestry. The more specific term of orthology delineates that orthologs arise by vertical descent from a single gene in the last common ancestor. This means that the concept of orthology is inherently hierarchical, because orthology is relative to the species radiation under consideration. And this gives rise to the concept of orthologous groups, which are defined as all genes descended from a single gene of the last common ancestor. So the last common ancestor 1 in this figure is the last common ancestor of just species A and species B. All genes descended from that single gene in last common ancestor 1 belong to a single orthologous group, A1, B1, and B2, where B1 and B2 are in the same species and are paralogs. Last common ancestor 2 encompasses all 4 species and all the evolutionary events that have happened since that last common ancestor. So that would include all the genes in the extant species, A1, B1, B2, C2, C3, D1, D2, and D3. Last common ancestor 3, however, there are 3 ancestral genes in this last common ancestor, with 1 loss in species C. So resolving orthology at this point in the phylogeny, we should have 2 separate groups, C2D2 and C3D3, and a singleton D1. So hopefully this explains the concept of hierarchy in orthology and how it's important when you're considering different sets of species from a much more fine-grained view of orthology when looking at more closely related species to much more inclusive orthologous groups when you go further back in time. Now, it's also important to mention what orthology is not. Orthology and paralogy are both concepts that are defined by purely evolutionary scenarios. So there is nothing in this definition that refers explicitly to gene function. So orthology is not equal to function. Nevertheless, because homology refers to common descent, generally, just as the sequences or the genes themselves are inherited, so too can their biological functions. Thus, orthology in many cases can be equivalent to function. And this, I point you again to the review from Kunin, where he talks about this crucial property of orthologs being both theoretically plausible and empirically supported, that they typically perform equivalent functions in the respective organisms. And not to forget the paralogs, just as in the case of orthology, the definition itself doesn't refer to function, but generally there are major functional connotations, as paralogs perform biologically distinct functions even if they are mechanistically related. So this is an important point to keep in mind, that the definitions of orthology and paralogy are purely based on evolutionary histories, but they do have implications for inferring gene functions. So having been through a little bit of some of the background in terms of the definitions of the terms and where they come from, now we can get to grips with some of the methodologies behind how we go about delineating orthology. For this, I point you to a chapter in a book in Methods in Molecular Biology called Inferring Orthology and Paralogy, where the authors go into some detail behind the main methods for delineating orthology and compare some of the major methods out there. Here we're just going to skim over some of the basic ideas. Orthology delineation, the methods behind it, can be separated into two main approaches: tree-based approaches and graph-based approaches. For tree-based approaches, involve a tree reconciliation step, where essentially we're trying to resolve the incongruences between the gene trees and the species trees by explaining them in terms of evolutionary events, speciation events, gene duplication events, and gene loss events. And for this, most methods rely on parsimony to come up with the most likely scenario that explains the incongruence between the gene tree and the known species tree with the fewest number of gene duplications and losses. And if you think about the first few slides where we were looking at the trees and we were looking at gene duplication events and gene loss events, that is what we were doing in our minds. We know the species tree and we're looking at the gene tree, and we are performing a reconciliation in order to define speciation, duplication, and loss events on the tree to identify orthologs and paralogs. Graph-based approaches start by constructing a graph of all pairwise gene homologies, where the genes are nodes on the graph, and these are connected by edges that reflect the evolutionary distances between pairs of genes or proteins. Then the clustering considers all of this pairwise information to build the orthologous groups. And you can see the similarities between the two in that we are trying to identify the relations between genes in extant species and reconstruct their phylogeny up to their last common ancestor. So now to go into a little bit of detail about how OrthoDB delineates orthology. OrthoDB would be classified as a graph-based approach. The first step is simply to select the longest protein coding transcript from genes with alternative transcripts, because we're defining orthology at the level of the gene, not at the level of alternative transcripts. The next step is to remove near identical proteins from each gene set, from each species. So we have the complete gene set, we filter out highly identical proteins, and we keep the representatives that go as input for clustering, while the filtered genes remain to be added later. The first major computational step is to perform the all-against-all Smith-Waterman pairwise sequence alignments. These are used to define the best reciprocal hits, or BRHs, between proteins from each pair of species in the analysis. So proteins in species A are compared to B and vice versa. And when the best match is reciprocal, this defines our core starting point, or our best reciprocal hit. Even if there are multiple hits from species A to species B, as in this case, so long as the best hit is reciprocal, we can still identify a best reciprocal hit to go into the clustering procedure. This falls down when the scores are very similar, or the best hit from A to B is not the best hit from B to A. And here we cannot define any best reciprocal hits. Moving on, these best reciprocal hits are used to start building best reciprocal hit triangles. So for each pair of best reciprocal hits, we start with the highest scoring ones and move down the list, looking at a third species and then asking the question of whether that triangle is closed also by a best reciprocal hit relationship, to form the core of a cluster. We continue moving down the list at increasing evolutionary distances, identify new best reciprocal hits, and check whether they close the triangle to join the growing cluster of orthologs. So this is continued with more and more evolutionary distant species as the cluster grows to form this core cluster made up of triangulated best reciprocal hits. The next step is to add best reciprocal hits that are connected to triangles, but which themselves do not form triangles. And here we increase the stringency to allow these new genes to join the growing clusters. So a best reciprocal hit that only forms a best reciprocal hit but not a triangle can be allowed to join the growing cluster. The next step is to add parallelize groups. These are within species homologs but are in different clusters. So if the within species homolog scores are better than the within cluster best reciprocal hit scores, then the parallelize cluster can be merged into the main cluster. And we'll try and explain this in the diagram. So here we have our core cluster, and we have another smaller cluster that contains 3 genes, but also from species A, B, and C. These 3 are homologous to the genes in the main cluster. And the scores are between A and A prime, and B and B prime, and C and C prime are better than the best reciprocal hit scores between A, B, C, and any of D, E, and F. Therefore, this triangulated A prime, B prime, C prime cluster can be merged into the main group. This is often much easier to think of in terms of a phylogenetic tree. So because we're looking for all genes descended from a single gene of the last common ancestor, the evolutionary event that we are seeing here is the duplication at the ancestor of species A, B, and C to create one set of A, B, and C, and another set of A prime, B prime, and C prime. But when you think of it in terms of a phylogenetic tree, this makes perfect sense why these genes should be merged into this cluster, because they originated from an event after the last common ancestor of all species under consideration. Finally, we look at all within species homologs that didn't get clustered. And if the within species homolog score is better than the within cluster best reciprocal hit scores, these singletons can be added to the cluster. We also add back those near identical proteins that were initially excluded from the clustering. So here we have homologs E prime and A prime that, because they are closer to the genes that are already members of this cluster, can be brought in to the final cluster. Now, of course, in reality, the data are much more complex. We do not have quite such beautiful links, and it's never so easy to determine when and where clusters should be merged. Here we have an example, a real-life example, of differential gene losses in 2 separate orthologous groups, one that encompasses the POP3 gene and one that includes the POP2 gene. Now, POP3 is missing from 10 vertebrate species, and POP2 is missing from 4 vertebrate species. This gives rise to all of these best reciprocal hit links between the 2 clusters. And so we have to prevent clusters from merging when the within cluster connectivity is much, much stronger than the between cluster connectivity. Now, these can arise from real biological events, because genes can be lost in different species, or they can arise from incomplete annotations or erroneous annotations in the gene models, which cause the pairwise data to be slightly messy. So in our wonderful ideal situation of fully connected graphs, we have missing links and incorrect links. And so we have to try and account for that with rule-based decisions on when and where different clusters should or shouldn't be merged to form the final orthologous groups. So to summarize, OrthoDB orthology delineation starts with defining all the pairwise relationships using all against all protein sequence alignments to delineate the best reciprocal hits between pairs of species. The next step then is to build the core clusters, which is done through a progressive triangulation of best reciprocal hits. Then finally, these core clusters are extended by adding the pair-only best reciprocal hits, and then adding the paralogous groups, and finally adding the paralogs to build the final orthologous groups. So we've covered some of the definitions, and we've looked at the methodologies, and in particular looking at the methodology behind OrthoDB as an example of a graph-based orthology delineation approach. And now we're going to look at in more detail at what OrthoDB offers to you as a researcher. Firstly, in terms of species coverage, OrthoDB covers now bacteria, archaea, viruses, and eukaryotes. Of course, I think most people on this call would be more interested in the eukaryota, and in particular the arthropoda, now covering 133 species of arthropods, with orthology defined at each major radiation on the species phylogeny. So here tracing back from Metazoa to Arthropoda to Insecta to Holometabula down to orders Hymenoptera, Diptera, etc. Access to the database is of course provided through the web browser, which we'll look at in a minute, but also through a JSON-based API, and we'll go through an example of that, with full data downloads available, and of course not to forget that you can download and install the orthology delineation software yourself and run it on your own data. In terms of i5k, the pilot project now sequenced 28 arthropod species, and these species have dramatically added to the diversity of the sampled lineages in OrthoDB, which were previously really dominated by the dipterans and the hymenopterans. We now have many more examples from some of the more basal insect lineages to really begin to sample these previously inaccessible regions of the phylogenetic tree. So to use OrthoDB in your research, you first have to go to orthodb.org, and you'll be greeted with the home page. The first thing is the text search, where you can simply type in a gene name or gene identifier or Go term or interpro ID to start looking for genes that are linked to that term. You can also search by sequence, so you can paste in a protein sequence of the gene of interest to identify the best match in OrthoDB. These are the two main entry points for browsing orthology data at OrthoDB. When performing the text searches, you can also apply phyloprofile filtering of the results. What this means is simply filtering your results to include orthologous groups that are only present in all species, or in more than 90% of species, or more than 80% of species, or those that are single copy in all, single copy in more than 90, or single copy in 80. So you can filter your initial search results to look for universal orthologs and single copy orthologs, if that's what you're looking for. Now, because we have so many species at OrthoDB, this can be rather overwhelming. But now with the species selector on the tree, you can really focus on the species that you're most interested in. Usually the species that you're studying, and then perhaps a couple of model organisms for which more functional information might be available. And to do this, you can select the species of interest from the expandable tree, or you can select nodes on the tree, which would then select all species that are children of that node, or you can use the search species by name function where you can start typing in the species, and the autocomplete will identify whether or not that species is already present in OrthoDB. As you select your species, the selection tree will expand to show the species that you have selected, marked with a tick here. And in the Species to Display panel on the top right, you will be shown a summary view of all the species that you have selected. Going back to this text search, you can then type in a term here using immunity, and the autocomplete will show the number of cached terms that match whatever you're typing. You'll note here also that the search at has defaulted to Endopterygota, so holometabula, because it automatically sets the search to the last common ancestor level of all the species that you have selected. But of course, you could use the drop-down menu to move further back in time and select more ancient last common ancestors. So in the case of this example of searching for immunity at the holometabula level, we get 10 orthologous groups returned. Here just shown in this summary panel. To start off with, at the top, you have the option to drag this bookmarklet into your bookmarks bar so that you can quickly and easily perform searches at this level on OrthoDB in the future. You have the option to download all the protein sequence data for all the results in FASTA format, or the option to download all the tab-delimited annotation data for all the genes in all the orthologous groups that were returned by your search. Moving back to the main results page, each bar starts with a super short summary information for the genes in that orthologous group, where such short information exists, because of course there are many orthologous groups with not enough annotation data to come up with a summary. You also have summary gene and species counts on the right-hand side, telling you how many genes are in the group and from how many species they come. And by clicking on the Expand button, you can then look at an individual orthologous group result in detail, here on this group of peptidoglycan recognition proteins. So clicking on that expands the detailed information for this PGRP orthologous group. Again, you can download FASTA formatted sequences or tab-delimited annotations, but this case just for the genes in this orthologous group, not for all the results as we saw previously. And you can collapse the view by clicking on that chevron. At the top, you have any functional descriptions for any of the genes included in this orthologous group, starting with, in this case, the gene ontology molecular functions and cellular component information, followed by interpro domains. Below that, you have evolutionary descriptions of this orthologous group. And on the right-hand side and all across the website, you will see these little question mark icons that you can click on that will take you to the help page to give further information about what these information are telling you. In this case, the phyletic profile gives you an idea of the universality of the genes in this orthologous group and how single copy or how multi-copy it is. So in this case, it's mostly multi-copy. The evolutionary rate is a measure of the sequence divergence of the proteins in this orthologous group. In this case, rather average, not very fast evolving, not very slow evolving. And the gene architecture panel gives you a canonical view of the structure of all the genes in this orthologous group, the median protein length and the median exon count. Scrolling down the results, you get to the orthologues by organism table, where you can view the individual genes in this orthologous group. Now here, we're just viewing the species that we selected. And just by clicking on that tick box, you can toggle between viewing all the species in this orthologous group or focusing just on your selected species. On the left here, we have the public gene identifiers for the genes in this orthologous group in this species. Then we have short descriptions, if available, for each of the genes, with these clickable chevrons indicating whether or not there's any further information, annotation information, available for each individual gene. On the right-hand side, we have information about the length in terms of amino acids, the number of exons in each of these genes, and any mapped interprodomains. Here, the mapped interprodomains just for Drosophila melanogaster in the order in which they appear in the protein. You'll notice here that some of the amino acid counts for each of these genes have exclamation marks either after or before. This is simply an indication that these genes are longer or shorter than the median protein length for the orthologs in this orthologous group, which can be a handy hint in some cases to identifying problems with gene annotations. Finally, down at the bottom, we have the sibling groups. That is a list of all orthologous groups at this level that are related to this orthologous group because they share common interprodomains. So to get more information about an individual gene, you would simply click on the chevrons to expand that annotation panel. Here, expanded for this particular gene, PGRP-SB1 from Drosophila melanogaster. Being from Drosophila melanogaster, there are many, many different sources of functional annotations available. The main one, of course, coming from Flybase, but also Ensemble, Uniprot, and many others. And the original search term that we searched for is highlighted here in yellow. So that brings me to the help page, of course. Wherever you see the question mark, you can click on that to get more information about where the annotations come from, what kinds of searches can be performed, and what is meant by some of these evolutionary descriptions of each orthologous group. So now for perhaps the more advanced user, perhaps wanting to download data on a much larger scale rather than simply browsing gene by gene for genes of a particular family or genes of particular interest for your biology of your specific organism, I'd like to walk through a simple example of using the application programming interface for OrthoDB. And to learn about all the different options available for the API, you can simply visit the API description page. Basically, this provides command line access to the data behind OrthoDB. And most of the queries will be returned in JSON format. Don't worry if you're not familiar about that. Go and find a bioinformatician and get them to explain it to you. And hopefully they'll be able to get out the large scale data that you're looking for from OrthoDB. So here's just a very simple example of programmatic data access using the API. Just one line query from the command line. The first part is a simple webget into the output file called myogs.txt, and this is followed by the URL that defines the query to OrthoDB. So in this URL, we are going to use the search mode. We're going to tell OrthoDB which level we would like to search at and which species to return. Now these level and species numbers correspond to the NCBI taxonomy identifiers. So if you know your bugs rather well, you'll know that 7070 on the end there is tribolium. And lastly, we'll define the query term. And again, we'll use the term immunity, just like we did when we were browsing the website. So running this command connects to OrthoDB and performs the query and saves the results into the file as you specified, myogs.txt. Looking at the results, this is what JSON format looks like. It's simply some messages about your query and a list of orthologous groups returned by your query. In this case, 10, because we searched at the holometabula node for the term immunity, just as we did when we were browsing. Now with a simple one-liner, here in this case using Perl, but of course you could use any programmatic language you like, we're going to loop through each of the orthologous groups that was returned by that first query that we stored in that file called myogs.txt, and perform a new query of OrthoDB, again using the webget for each group, and this time performing a tab search to get annotation information for each of the genes from each of the species in each of these 10 orthologous groups. And in this case, we're going to add the option long equals 1 at the end, which allows us to retrieve the protein sequences at the same time. Again, we connect to OrthoDB, we send the request, and it's saved to the file, in this case with the name of the orthologous group, .txt. Looking in that file, we have the public orthologous group ID, we have the name of the orthologous group, if it does have a name, we have the level at which it was defined, the species from which this gene comes, the name of it, the internal protein ID, public gene ID, a description if available, and finally the sequence. So you can see how this is a really flexible way to gain access to data from OrthoDB. Now I'd like to move on to a relatively new feature in OrthoDB version 9, which allows you to perform comparative analysis with your own data. So clicking on the comparative charts at the top, you'll be greeted with a login or create your own account page. So once you've done that, you will have access to an additional tab called Own Data and Mapping Facilities. And here I'm going to use an example of a very newly published aphid genome, and we're going to map the proteins predicted in this genome to OrthoDB. So the first thing you need to do is upload your file, in this case faster formatted sequences of all the proteins predicted from this aphid species. Now you will please have to make sure that your data are in proper FASTA format, otherwise it will be rejected. And remember, just like at the very beginning of our orthology delineation process, we selected just one protein per gene in the case of alternative transcripts. So you will need to do some pre-processing of your data before you upload it for performing your comparative analyses. Once your data are uploaded, then you can request to perform orthologous group mapping against choosing 1 to 5 comparison species. Note here that the node to which you want to map your data is automatically selected as the last common ancestor of all the species that you have chosen to use as your anchor species for mapping, in this case, Insector. And then we click Run Analysis, and in a little while you should get an email saying that your job has started. Now please bear in mind that this is an all-against-all search of all your protein sequences against up to 5 species in OrthoDB. So this can take quite some time. This is not a Google search. You probably need to go away and come back the following day, especially if there's heavy traffic. So at the moment, if it's really quiet, you might get your results back by the end of the day. But if you all go online after this webinar and try to do it at the same time, then you will be waiting quite some time. Once it's done, you should receive an update on the final progress of your mapping job by email, and then you can come back to the website to view your results. You will note, though, that once you've started your mapping, the species that you've added will now appear on the species selector tree with this little symbol indicating that it's a user-provided species. So once it's done, then you can either download the full results of your gene IDs mapped to OrthoDB orthologous group IDs, or you can browse OrthoDB just as you would normally, but now also including your newly mapped species. So the data download simply provides you with the orthologous group ID and your input gene IDs and some information about the scores for their mapping. But more importantly, perhaps, is you can immediately start browsing on OrthoDB the results for your species that has just been mapped right next to the results from the existing species in OrthoDB. Of course, annotations are not provided, and number of exons and protein lengths and interpro domains are not provided for your species. But you can very quickly begin to browse for specific genes of interest using, for example, model species to guide you to find them in your species. The comparative analysis continues with the comparative charts options, where you can select up to 20 species on OrthoDB to produce an orthology profile for your selected species. So just as you would select the species for browsing OrthoDB, you can select the species for creating a comparative analysis orthology chart. This will take some time to run. In this case, I've selected, I think, 18 different species, and the result classifies all orthologs from all of these species according to their single copiness and their universality. And this graph is completely configurable. So if you use the Configure Chart panel, you can change it and choose the most disgusting colors you like. And simply export it, and it's ready to go in your manuscript. Now, of course, having just performed a mapping of your particular species, in this case of that newly published aphid genome, you can see how your orthologs compare to various other species in OrthoDB. Once the mapping is complete, you can perform a comparative analysis chart building with your own data and any other representative species. I will add here, though, that when you are selecting your species for orthology comparison charts, please be mindful that some genome projects may not yet be published. So please respect the conventions and do not use data from unpublished genome projects without the explicit permission from the data producers themselves. Lastly, you may have noticed, as well as being able to perform orthology mapping, you can also perform a BUSCO assessment with benchmarking universal single copy orthologs. At the moment, this is just for protein sets, not for genomes. BUSCOs, for those of you who are not so familiar, are selected from orthologous groups with genes that are found in the majority of species as single copy orthologs. This leads to an evolutionary expectation that they should be found in any newly sequenced and annotated genome, and they should be found as single copy orthologs. And the BUSCO tool implements quality assessments in terms of gene content completeness for genome assemblies, annotated gene sets, and assembled transcriptomes. Through OrthoDB, we provide the ability to assess just your annotated gene sets for the moment. The bonus features of these benchmarking universal single copy orthologs is, of course, that they also provide excellent markers for phylogenomic studies and are a really good set to use for gene predictor training. So coming back to the results, here again for the recently published aphid genome, I highlight a statement where they claim that this assembly shows high completeness with almost 20,000 predicted genes, 92% of transcripts that map back to the contigs, and good N50 continuity. However, they did not perform any assessment of completeness in terms of gene content. So by running their protein set through OrthoDB BUSCO assessment, we can quickly come up with a quantification of the completeness in terms of gene content. And here, in fact, they don't score too badly, with 91% complete. So we've had a little look at what we mean by orthology and the underlying definitions. We've been through some of the methodologies, focusing mainly on how OrthoDB delineates orthologs. And then we've had a brief look at how you might search OrthoDB, how you might use the OrthoDB API, and how you might use some of the comparative analysis features now offered by the latest incarnation of OrthoDB. So to finish with, I'd like to briefly discuss why we actually need to delineate orthology. The first main reason is to trace the evolutionary histories of all genes in extant species from those that are widely conserved across many species down to those that are much more lineage-specific or even species-specific. The second reason comes back to what we were discussing about how orthology can be equated with function, as it provides the best hypothesis on gene function because it is informed by the evolutionary histories of the genes. So as orthologs share a common ancestry, they can be considered to be equivalent genes in different species. And therefore, any hypothesis that they share a common function is a relatively reasonable best guess assumption. And as Kunin comments on in much more detail in his review, the huge majority of genes in sequenced genomes today will never be studied in great deal experimentally. So for most genomes, the transfer of functional information between orthologs is really the only means of such detailed functional characterization, and hence the importance and the need for orthology delineation. So to finish with, I'd like to remind everyone that if you have, or if you will, use OrthoDB in any of your research, please remember to cite the relevant publication. In order to keep our database going and to keep up with the flood of data, we need citations in order to get funding. So please remember to cite the resources. And this goes for all genomics resources. And with that, I would direct you to email us at support@orthodb.org with any questions, and finish with the whole team involved here at OrthoDB. It's not just me. Many of us working on this project for many years. And I'd be happy to hopefully take some of your questions in the last 10 minutes. Thank you.
Thank you, Rob. I'm going to go ahead and send out, again, the information on how to submit your questions. I've got a couple here to go ahead and get started with. The first is, what is the best way to do a comparative visualization of positions of orthologs in the genomes of fruit flies and mosquitoes?
So for that, you would need some sort of synteny visualization tool. I can't think of one off the top of my head, but you could use the data from OrthoDB in terms of which are the equivalent genes in different species and feed that into a synteny visualization tool. So currently OrthoDB doesn't offer any synteny visualization, but you can use the underlying data to feed that into a visualization tool, definitely.
Wonderful. Okay. The next question, for researchers interested in analyzing their own data using the new web service for comparative analysis that you just introduced, are there recommendations for minimum quality of the protein set?
So I guess there the answer would be first to run Busco to see how complete your protein annotation set is before you then attempt to do the mapping. And just like with any other comparative analysis, you want it to be as complete as possible. But if you're fishing for perhaps a conserved subset of single copy orthologs, then the results from the analysis will still be valid, but of course you will not have a complete set.
Wonderful. All right. What are the default similarity cutoffs for calling sequences orthologs?
To define the best reciprocal hits, that's where we use the cutoff. And there we have on the Smith-Waterman alignment an e-value cutoff for the best reciprocal hits of 1 to the -3. And then for when these best reciprocal hit pairs join triangulated clusters, we increase that threshold to 1e-6. So it's an expectation cutoff, not a similarity cutoff. And they must meet the criteria of being best reciprocal hits.
How does OrthoDB find and choose the gene sets for each organism, given that multiple gene sets might exist between NCBI and community data sets?
Yeah, so this is a perennial problem. generally, we try to listen to the community and see which one that they are primarily working with. So it's a push and a pull, basically. People who get in touch with us to try and put forward the newest, best annotation, especially if they can convince us that it is better, then we'll definitely go with that. There's also considerations of being able to link back to the source databases. So if those source databases are more stable and more reliable and more accessible, then we're probably more likely to go with them, right? So when you're browsing OrthoDB, if you want to see where the annotation came from, we would like to be able to link directly to the source database. So for example, if you're looking at fly genes, you should be able to click straight through to view the full information for that gene at Flybase.
Does OrthoDB provide information which unpublished data is used?
That is rather a headache to keep track of all of that for 500 eukaryotic organisms, so no, we don't. I would say to anyone who's looking to do comparative analysis and use the species in OrthoDB, you probably are the best placed person to know about what's published in your field, right? So you need to make that call. And by all means, use all the data available for your own analyses. But if you plan to publish anything, then please be aware and make sure that any species you choose for your comparative analyses are either already published or you have the explicit permission to use these data in such comparative analysis.
So next question, regarding the clustering pipeline, has it been benchmarked against other ortho databases clustering approaches? If so, where are the strengths and weaknesses of this OrthoDB clustering?
Yeah, so there are a couple of benchmarking studies out there. I didn't include any of them here, but you can email me afterwards for those references, comparing some of the major different pipelines out there for orthology delineation. I would say one of the main differences between graph-based approaches and tree-based approaches is coping with the flood of data, because the tree-based approaches need to perform a full multiple sequence alignment in order to then build the gene tree, which they can then reconcile with the species tree. And as more and more genomes are being sequenced, those multiple sequence alignments are getting bigger and longer and taking longer to compute. And of course, the phylogenetic trees that you get out of them at the end are getting more and more difficult, right? So this is a strength perhaps of the pairwise approach and the graph-based clustering approach, is that it's much easier to keep up with all of the new data that are arriving. Hence being able to map new species to existing clusters, because we are basing the orthology assignments on pairwise information. Whereas if you were trying to align a whole set of sequences, that would be much more computationally intensive. But yeah, do remind me about that afterwards, and I'll send out a couple of references that go into some detail with various different measures of comparing the results from different orthology delineation approaches. Okay.
In case someone wanted to make sure their data would be included in the next release, what time frame do you have, or at what point in the year do you freeze everything in order to do your next release?
Traditionally, it's been January, but these things change. And as the data grow bigger and bigger and bigger, we actually have to adapt our strategy rather. So one of the main points of making this orthology mapping into a much more streamlined pipeline is that in a way any new species can be added progressively to the current version of OrthoDB. You can add your data, and when you're happy with it, you can make it public. So in that way, species can be added one at a time almost to the current instance of OrthoDB before we then go on to the complete reassessment of orthology, probably annually.
Okay, and I guess maybe a final question here. Many of the genomes that people are sequencing right now might be considered draft quality and therefore won't have a complete annotated gene set, and like you mentioned, that can be a difficulty. Do you have a threshold for genome or gene set completeness in order to include that species in OrthoDB?
At the moment, the way we're dealing with that is we're using Busco assessments to choose the best quality annotations from all species from all clades. Those that don't score so well, we leave out of the clustering procedure and we map them on to the orthologous groups at the end. Because as you can imagine, the less complete annotations interfere with those triangulation of best reciprocal hits and can confuse the orthology delineation process. So in fact, we're already doing that. And when you browse OrthoDB, when you're looking at the tree, you can see species that are clustered, and you can see with a little symbol species that have been mapped. And these are generally species with lower completeness scores. So if you're concerned about that, it's not a problem. Your species would just be mapped to the core defined clusters rather than being included during the clustering procedure.
Wonderful. Rob, I want to thank you. We've reached the end of the hour, and so thank you to Rob and thank you to everyone that has called in. This has really been a great presentation and a great discussion. Our next webinar will be Wednesday, March 1st, and that will be with a representative from Bionano. And we will do our best to we've got the slides up already, and we'll do our best to get the video up as soon as we can. So thank you all again, and have a wonderful day.
Thanks, everyone. Goodbye.